Colleagues here in King's Conference Centre, enormous welcome. Colleagues around the world, an enormous welcome. So I'm so delighted that tonight not only are we able to share the great thoughts and stories and journeys that Neil, Jim and Anne will describe to us with those in the conference centre, but equally live streaming in a truly 21st century way, takes the words of wisdom from the three of you, not only to the world this evening, but for eternity. And that can't be bad, because I think it's an incredibly exciting night for the university. In a week after we learnt that fully 7 or 8 of our research areas feature right in the top 100 of global science, including the 3 areas here, including also our work in engineering, but just demonstrating the breadth of this university. The area with our highest ranking was theology, a university of incredible breadth, but a university with real strength everywhere. And while you know I would be delighted to go on at some length, I'm not going to, because tonight we're here really just to look at some of our real stars. And one of the absolute foci of, if you like, excellence is fellowships of the learned academies. Just 2 weeks ago, we learnt that this year we had more fellows elected to the Royal Society of Edinburgh than any other university in Scotland, We have a significant number of fellows of the British Academy. But we hadn't had so many fellows of the Royal Society. And in the spirit of London buses, we hadn't had any for a while, and then 3 came along all at the same time. 3 came along all at the same time, and only 2 universities in central England can match that kind of record in 2016. How about that? We sit here. We're not very good in the Northeast at, if you like, celebrating and saying how good we are. Tonight is an opportunity to do that. I'm incredibly privileged as principal to be able to share with you this evening, to have scholars of the quality of Neil, Jim and Anne to speak to us this evening. But also I think just to mention that one of the things we talk about so often is creating an environment for careers to flourish. The 3 people we're going to hear about tonight didn't, if you like, appear at the writing of a checkbook at the end of their career. I have to tell you, I haven't got a checkbook to write with. But were able to develop their careers here over a period of time and go to the great glory that we celebrate. Now, Neil Gow is going to kick off. Firstly, I would tell you all about his CV, but then that would be to give his talk. So, Neil, I won't do that, simply to say we're absolutely privileged to have you and invite you to talk to us.
Thanks very much. Well, thank you very much, Principal. And thank you very much for coming along and to hear something about a series of stories with actually a common denominator, because it's a great privilege for me to talk today, sharing a stage with 2 people who have been colleagues for all of my adult professional life. We were in the same department for much of our time in Marischal College, and we kind of evolved in slightly different directions, but what we've all had is a love of microbiology, the study of microbes. And Anne and Jim, their flavour of microbiology happens to be for bacteria, and I am somebody who works on fungi. And so there's my talk, and I'm part of, you'll see at the bottom of my slide, the Aberdeen Fungal Group, and that represents the largest group of people in the UK, to my knowledge at least, that work on fungal systems. There's about 80 scientists, if you include all the PhDs and members of staff who work on this. Now, we're very kind of proud of the work we do, I guess, but we know that when we face you, the general public, that there are amongst you people who are less clear why on earth your hard-earned tax pounds should be paying for people to study a fungus. And we know that sometimes when we're invited to dinner parties, that when you declare what your particular specialist is, that isn't always a conversation starter. But I'm going to start by telling you why you should feel that some of your money, which does feed into mycology through your taxation, is well spent. And the first thing I'll just say is that I'm going to hazard a statement, an assertion that every one of you today and every day of your lives will have used or benefited from a fungal product. And here's just a list of all the things that we get from fungi: antibiotics, drugs, and various chemicals and enzymes, food products, as well, of course, as all of the products which we have in the beer, wine, sake industry, some of which are very important locally, of course. And a second reason why fungi are worth studying is evidenced by the fact that at least nearly a dozen scientists internationally have obtained Nobel Prizes for working on fungal systems. And you can see some of the people there, a variety of fungal species which are the organisms of choice that they worked on. And so there must be something pretty interesting about working in a fungus that this has generated this degree of academic activity at the very, very highest level. But for most of you, or for most people who you are at your dinner party, you tell me you work on a fungus, this is the image that they have, and that's quite appropriate. Fungi are a natural part of our ecosystem. What most people don't realize is that fungi are a very dominant part of our ecosystem. There are more than 5 times the number of fungal species than there are plants, for example. And if, as a crude estimate, if you estimate the total number of animal and plant species and put them all together, that's about the number of fungal species there are in the world, about 5 million different species. And some of them are absolutely gorgeous and beautiful. This is a relatively recent discovery of a fungus. It's a coral fungus. It looks like a piece of coral. Discovered, would you believe it, in Iceland in the last couple of years ago. Very beautiful organisms, a part of this huge and relatively unexplored bit of biodiversity in the world. Now, when you look at a fungus, you might say, well, that's not really a microorganism. What you see is what you're seeing above the ground. But underneath these, and if I run this movie in which background music, these are fungal cells. This is what supports the growth of a fungus. This is what you see underneath the soil. And these beautiful structures, this network of a fungus is called a mycelium. And inside you can see structures. These are nuclei that you're looking around, racing around the cell, very dynamic process. These cells are growing and dividing. And interestingly enough, the processes that they use is really highly conserved in nature. So what's happening in there, to a large extent, is what's happening inside your own cells. And that's one of the reasons why there are so many Nobel Prizes for people studying fungal organisms, which of course are very easy to study and very good genetics for. And they grow in this way, or they grow as yeast, little rounded cells, and they do so and reproduce their life forms in a way that we can understand not only about the fungus and about that biodiversity, but of course directly about our own selves. And here's the microscopic view, equal levels of beauty, extremely beautiful organisms. So there's 3 reasons why you should study fungi. Now, my branch of this is a slightly sinister side of mycology. It's the dark side of mycology, if you want. Fungi are organisms which grow extremely fast. This is a giant jelly petri dish, and what you're seeing evolving here actually took about 5 days to develop. And it's spelling out the words in microbial biomass killer fungus. And this comes from an exhibition that we ran last year in the Royal Society. And some of these organisms, these are actually safe varieties, because we were showing a lot of people these petri dishes at that exhibition. But these are organisms, the fungi are organisms which also can cause really serious human disease. And that's what my speciality actually is. Now, for those of you who have said, well, I've heard about athlete's foot, that's about it. That's what most people have heard about. We put together a movie for this, and I'm going to sit down and have a glass of water. This movie takes 2 minutes to run, but it pretty much summarizes why you should be a medical mycologist. And it has the voice of one of our rising stars up at Forester Hill, Duncan Wilson, narrating this. So I'll let this run, and then I'll tell you a bit more.
Fungi play many important roles in the world's ecosystems, and most of us actually carry harmless fungal colonizers on our skin and in our digestive tracts. But there is a dark side to fungi. Every year, more than 1 million people die from fungal infections. That's more than those killed by malaria or breast cancer, and almost as many as those claimed by HIV or tuberculosis. On top of this staggering and hugely underappreciated death toll, fungi cause at least 100 million mucosal infections and 1 billion skin infections every year. This is a huge problem, compounded by a complete lack of vaccines against fungal infections, a limited arsenal of antifungal drugs available to doctors, and often unreliable diagnostics. The problem is increasing with drug-resistant fungi that may have evolved from crops that are being treated with agricultural fungicides. So why, in the face of this global disease crisis, haven't you heard of these hidden killers, when others, like MRSA, SARS, and Ebola, regularly make headline news? Thankfully, a small but highly active research community are fighting back against the killer fungus. Scientists in the UK, along with our international partners, are making great strides towards understanding how these fungi cause disease. In doing so, we are coming up with new methods to detect these infections and provide new therapeutics and disease interventions. We now understand how fungi are recognised by our immune system, paving the way for treatments to boost immune responses and combat fungal disease. Our greater understanding of how fungi infect the human body is allowing us to design new drugs that selectively target fungal-specific structures, such as its cell wall. Antibodies are being developed that can be used to treat infections and to diagnose these infections early, which will greatly improve patient survival. We're also directly involved in training the next generation of scientists and doctors to tackle fungal disease, not only here in the UK, but in developing countries such as those in Africa, Asia, and South America, where the fungal death toll is especially high. Together, our research will help us to win the battle against the killer fungus.
Okay, so that was the strapline, and this strapline, these take-home messages are things which lots of people, including many other microbiologists, have been less aware of. These are organisms which cause problems in a real grand scale, so benchmarking them, everybody's heard of malaria, but there are more than twice the number of people who are dying from malaria than fungal infections than malaria, and the death toll is similar to things like tuberculosis and HIV, which people have heard of, and more than double that, which cause the annual death toll in breast cancer. So really extremely serious diseases and underappreciated. And in fact, the people we had to persuade were not just, and to inform, were not just the general public, were actually the research councils themselves, because these numbers are the numbers which we got directly from the Medical Research Council, the Wellcome Trust, NIH and US and EC, about 2 and 2.5% of their total budget. When it's something to do with microbiology, something to do with infectious biology, that was the amount that went to something which had anything to do with medical mycology, a tiny amount. Reflecting not the fact that we don't know how to write good grants, reflecting the fact that there was very few people studying these organisms. Now, I'm very proud to say, of the successes we've had, 2 of the biggest awards that have ever been awarded in this field have both come to Aberdeen. So one of them is this strategic award on the left-hand side here, the Medical Mycology, Medical Mycology and Fungal Immunology, a strategic award, and that really places Aberdeen as a very sort of elliptical hub of activity. We coordinate the research across 12 universities in the UK. We are training a new generation of medical mycologists, not just in the UK, clinically and basic, but also in countries of low and middle income countries where there's a huge requirement for this expertise. And very recently, under the guidance of my colleague and friend, Professor Gordon Brown, we became an MRC, Medical Research Council Centre for Medical Mycology, the first in our field. So we're very pleased about that, and that gives us new opportunities to do more. So what do I do in this microcosm? So the organism I mainly study is this fungus. It's a split personality fungus, which most of us have. If you're typical of a patient group or a group of normal people, about 50% plus of you have got Candida in you, and don't have any symptoms of disease. It's sometimes known as a thrush fungus. And you can see by the images on the other side that this is a yeast. You can see the yeast on the top of this, up here, and it invades into epithelia and causes disease. And it causes disease in very large numbers of people. For women, there are 400 million women a year estimated to get 4 episodes of thrush every year, a miserable sentence. This affects children, it affects people with HIV, and for about 250,000 to 400,000 people, these are people with severe immune defects or trauma victims, they get this organism in their bloodstream, and that's extremely serious. And even a well-informed, fully armed clinician will only save about 60%, so a 40% death toll in that patient group. So what I do, everybody's got to have a point of leverage in the system. My group mainly works on the wall of the fungus. So I emphasize inside the fungus, the biochemistry of fungus is just like ours. They're very, very similar in many ways. The wall is what makes a fungus very different, and that's very useful and a very important thing to study, because the wall is made of molecules which are only found in fungi, not in humans. And all of these molecules you can see in this wall, this image of the wall, we've studied the genes and the enzymes which assemble that wall and make it into this process, and try to find exactly where goes what inside the wall. And why we should do that? Well, I'm going to give you 2 little vignettes of why we should do that. And one reason is because this should be a point of attack, where if we attack the wall, which is specific to the fungus, we should be able to kill the fungus. And secondly, we'll show you a new project which relates to a second novel way of detecting and attacking the wall using human antibodies. So there's a little image up there on top right. Why, you're asking, why don't we have just an antibiotic? Penicillin works against bacteria. Penicillin attacks the wall of bacteria, and it kills them very effectively, broad spectrum. Why doesn't this happen for a fungus? Well, you'll see here a couple of movies. Here's an aspergillus, which is a very difficult organism to eradicate. This is an antibiotic which attacks the wall. And if you look at this movie, initially all looks good. The cells are lysing and broken open, they're dead, but the fungus is recovering. And we've discovered this mechanism and worked out exactly how the mechanism works. And the other movie is a thrush fungus, and you'll see that the amount of blue fluorescence in this picture gets more and more as the cells are exposed to the drug. These cells are not dying, they're modifying their walls to make them resistant to the presence of the antibiotic. And this process is something that bacteria cannot do. So these are extremely difficult organisms to work with. So it follows logically, if you know what the rescue pathway is, you can design drugs against the rescue pathway. And that's what we're screening for now. So we could have a combination of drugs, one to attack the wall and one for the fungus to stop doing something about that. Now, another way forward might be to take advantage of what our bodies do already. So you've had a cold, and you know when you've had a cold that you've recovered from that cold. It may have taken you longer than you would like to recover from your cold, but during that time your body's doing something, your immune system's being activated. And similarly, the body recognizes fungal cells and tries to do something about it, but the problem is it's basically too little and too late. But can we help the body to do more about that? So this is a project which is the project of a very talented PhD student who's now a postdoc in my lab. And what Fiona Rudkin did was she started off with some blood cells from patients who'd recovered from a fungal infection. So we knew that they had antibodies against the fungus. She then took individual cells, these are B cells, the factory cells for antibodies, and took from an individual cell the genes and amplified the genes for the antibody, and then used those genes to place them in a factory cell where we could make bucket loads of antibody in large quantities, very pure. And then we could screen those antibodies against targets against the wall and use those to do useful things, diagnostics and drugs. So here's a picture of one of Fiona's antibodies, and you can remember that picture of the wall, which is frilly on the outside, and then it has this inner line. I can't really see that, hold on. And you can see it's binding here to the wall and nothing else. And we know that it doesn't bind at all to human cells, so it's perfectly safe. And it doesn't bind to other types of microbes, and it does bind to Candida. So there's the thrush fungus, and the yellow stain here is exactly where this particular antibody binds, and Fiona's made nearly 20 of these antibodies, which all bind to different things. And they're great. They work in all sorts of different ways. We can do useful things, so I'm now going to show you a couple of her movies in which we're having human white cells. These are the phagocytes which gobble up bacteria and other microbes when they enter our body and try and kill them. On the left-hand side, you'll see a movie of the Candida cells which are coloured in green. And the right-hand side, exactly the same thing, but with one of Fiona's antibodies to coat the fungus, and you'll see that those phagocytes, those macrophages, do much better. So just watch how quickly they're identified, grabbed, and taken up inside the macrophages. So you can see on the left-hand side it's happening. On the right-hand side, those are very, very hungry macrophages, and sayonara, those bugs are gone in much, much more quick time. And when we use these antibodies in a model of infection, they protect the animal from an infection. So these are potentially very useful. We can use them both to diagnose the presence of a fungus, and actually one of our antibodies has already been licensed to a diagnostic company. We hope it's going to be on the shelf in a couple of years, and we look forward to trying to use these antibodies as novel therapeutics to help our body do better in the future against this type of infection, which kills so many people. So if you want to know a bit more about this, and you want to play around with this, we also made a couple of games which you can download just now if you want, although I'd wait until the end of Anne's talk if I was you. And one's about antibiotics, this one here, and this one's about immunology, and you'll get a lot more information just by playing along with these games. These are very popular. At least it's the only thing that my kids have ever thought I've ever done which is of any use whatsoever. And if you're interested in this, you can't go to that exhibition which was in London, although we're trying to run it again in Manchester this year. But you can come to this exhibition, which is the Kingdom of Fungi exhibition, which is down at the Aberdeen Science Centre, and it is still available, and some of the movies I've shown you run there on a daily basis, as well as those games. So I hope I'll leave you with this image that although fungi are incredibly useful organisms, absolutely central to biology, they can be really horrible, but they're still very beautiful. So I'll use this picture of Candida to say, that's me. And I'll just finish by showing you one more picture, the key one, because this work is teams of people working together. We have a fabulous team. This is the Aberdeen Fungal Group staff that we work with, and many, many students and collaborators around the world which really enjoy working to try and make a difference in this area. And thank you very much for your attention.
Neil, that was absolutely brilliant. And I think, for me, one of the absolutely key things of all research stars is how brilliant they are at communication. So, Neil, thank you very much. You said that's all from you, but my job is to make the timing right. So we've got time for one or two questions. If anyone would like to ask a question, to do so, you do as that gentleman there has so effectively done. Press the button and a little red light comes on your microphone. Sir.
Great talk, Neil. Thank you very much. I just wondered whether the antibodies that your student isolated, do you know what they are? Are they recognizing a range of structures on the cell wall of the fungus, or are they homing in on quite similar things?
Sorry, so this works. Yes, thanks very much. Yeah, well, we do know what they raise. We raise them in two different ways. We took one fixed target and said, right, we want some antibodies to that. And we started off with particular proteins which we'd cloned and expressed, and we raised them to just that. And they worked very well. And then we also did another set of experiments where we raised them randomly to the surface, and we have a whole series of targets there, and we've now drilled down to find out what those targets are. And that's very useful because we now have a range of antibodies which will just detect one type of candida or many types of candida. They're useful for one type of application. Or we want them just to recognize Candida albicans and nothing else, and we can do that too. So this is I love this technology because it's very plastic and you can use it for all sorts of different things, and all of those things really work very well.
Sir.
Thank you very much. That was very interesting. Are there similar effects such as biofilms in the fungal world where there's actually sort of biodiversity and ecology going on in the body that makes this much harder to treat?
So Candida does form a very effective biofilms, and it chooses to do so on things which we really find which are essential for clinicians trying to save patients. For example, on lines, surgical devices, heart valves, anything man-made which goes in seems to be an excellent substrate for a fungus to grow on. They tend not to be a bit like the biofilms which you have naturally, which are usually very diverse. They usually build up a biofilm which in a sterile site, it depends on what's around. So there's usually not a lot of different bugs, but they can be Candida and a bacterium at the same time. And once they're there, they're really difficult to dislodge. And the effectiveness of antibiotics is much less. It's difficult for them to penetrate in through the biofilm. And often this means that the clinical person has forced to remove that line or change that device, which they don't want to do in a very sick patient. So biofilms are a real problem with fungal infections.
One final question. Please, sir.
Thank you. Do fungi attack one another? And could that be used as a source of medicine?
So in the natural soils, they do. There are fungi which attack other fungi. They're called mycoparasites, and they are used to a limited extent in natural settings, agriculturally. So you can use one to control another. And for example, in the Aberdeenshire area, one of the diseases caused by infections of pine trees at the seedling stage, a very effective form of biocontrol using one fungus to attack another is used here. And that works really better than the chemical options which are available to attack the fungus. So not so much in medicine, and in fact, using one bug to attack another bug, particularly viruses, bacteria viruses, this is something that's relatively unexplored, but so far not very successful. And there are not very many viruses which attack fungi, so that's one of the options which we don't have, unfortunately.
Thank you very much. Neil, thank you very, very much. I should add that Neil and his colleagues in August of this year will open the first University of Aberdeen laboratory in Africa to actually bring, if you like, the work so much closer to where so many of these deaths are happening. Neil, thank you for that inspirational talk. Colleagues, shall we thank Neil again? And now that moves me on to Jim Prosser. Jim will now describe his work.
Okay.
Great.
Right, well, thank you very much. And put my first slide up. Okay, so I'm a soil microbiologist, so I want to talk to you about soil microbes. Title there, Soil Microbes, So Small, So Many, So Diverse, and So What? And we were asked to reflect on some of the particular aspects of Aberdeen that have struck us over the years. And I suppose for me, the thing that, if I think back, the most important thing are people. People I've met in Aberdeen. And some of them are in the audience today. There are too many to mention, but there's a few that I feel I have to. And two are on the first slide. Alan Hamilton, who started the department of microbiology in Aberdeen, he's the one on the left. And Graham Gooday, who was his first appointment, who started a year later. And they were both here when I came in 1978. Graham looks as though he's just about to have his lunch there. I think it's a graduation reception. And they, for me, were inspirational and enormously wonderful examples of how to do science, how to teach in universities, how to do research, and so on. And I owe, and I suspect Neil and Anne will say as well, that they owe both of these people a great debt. Now, I want to talk to you about soil microorganisms, and I want to start off with an analogy. So here we have a planet, our planet, and on our planet there are around about 7 billion people. And then here we have a gram of soil. And within that soil, there are about 7 billion bacteria. I didn't spend a lot of time making films, but I'd spent a lot of time getting some props. So that's what a gram of soil looks like. Okay, it's not very much, but within that gram of soil, there are 7 billion bacteria. So as many bacteria in a gram of soil as there are people on the planet. Now, that sounds amazing, a massive number, but really what it means is bacteria are small. And in fact, the proportion of the planet covered by us is round about the same as the proportional of the surface area in a gram of clay soil covered by 7 billion bacteria. So if you actually look at the bacteria in the soil, you'll see them here as pinpoints of light stained on a soil section. You will see on the top figure there, and the bacteria, that's the top soil, and beneath that is the subsoil. The bacteria, although there's a lot of them there, they're actually separated by many long distances compared to their cell lengths. And their cell lengths are about a micron, a millionth of a meter. So they're abundant there, but what's much more important is that they have a lot of mass. And we get upset because people don't realize how important bacteria are in the soil. But if you look at if you drove past that field and saw all those sheep, you would think that those sheep were quite important for that environment. Well, the mass of bacteria in that soil is equivalent to the mass of those sheep. And if you, in some grassland soils which have high microbial biomass, it's more like 2,000 sheep, the equivalent to the mass of 2,000 sheep per acre or per hectare. So that's a serious amount of biomass there. And not only is the mass high, their activity is high, and their activity is very diverse and very important. So bacteria are responsible for creation of the soil in the first place, and then they're responsible for turning over nutrients, maintaining soil structure, and maintaining nutrients for plants. And they do bad things as well. Some of them can cause plant disease, but they are necessary for agriculture, even though you can't see them. Another thing about soil is it's very complicated. If we use X-rays to look at soil, you see this very complex structure. Lots of micropores, microenvironments, if you like, in which the bacteria live. Very varied in chemical composition and physical chemical characteristics. So there's a lot of variety there for microorganisms. So the soil microbial community is the most diverse community that you see on the planet. So this sort of thing makes it difficult for us when we want to study soil microbes, because we need to study them at this small scale, and that's very difficult technically, but it also makes this really exciting, because we've not had the proper techniques in the past to study these organisms, and now we do, and what we're discovering is amazing. And also, what they're doing is really important. Now, there's one group of organisms I'm particularly interested in, and these are organisms that are responsible for the first step in soil nitrification. Now, when a farmer adds fertilizer to the soil, he's usually adding ammonium fertilizers. So what happens is he puts ammonium into the soil, and that ammonia gets oxidized to nitrite and then oxidized to nitrate. And that's fine, because both ammonium and nitrate can be used by plants. But if it rains, and it does around here, then what happens is two things. First of all, the nitrate is washed out of the soil, and that leads to losses of tens of billions of pounds worth of fertilizer globally on an annual basis. So it's an enormous economic cost, this process of ammonia oxidation, this first step in nitrification here. Also, when it rains, oxygen is used up in the soil, and other processes occur. And in particular, the nitrate is converted into nitrous oxide, which is a greenhouse gas. So as well as causing this big economic problem, these organisms also cause an environmental problem, producing this greenhouse gas. Now, we're interested in this first step, where ammonia is converted into nitrite, and that's carried out by these bacteria here. And you see a number of pictures there. All of those cells are 1 or 2 microns long, different shapes, spiral-shaped, circular or spherical, rod-shaped, and so on. And in fact, the very first ammonia oxidizing bacterium was isolated by this guy here, a great name, Percy Faraday Franklin. Don't you wish you were called Percy Faraday Franklin? And he wasn't from Aberdeen, but he was nearly from Aberdeen. He was at the University of Dundee. And he isolated the first ammonia oxidizers with his wife, the papers by Franklin and Franklin, but of course, you can only find pictures of him, not his wife, unfortunately. Anyway, these are the organisms that carry out this process, and they have this enzyme, ammonium monooxygenase, which carries out the very first step in ammonia oxidation, and, because I might mention it later, importantly, they have a gene which encodes for that enzyme, which is called the AMOA gene. Okay, now, talked about the organisms. How do we study microbial diversity? How do we study the communities of microbes in the soil? Well, traditionally what we did, and traditionally means until 25 years ago, what we did was to grow the organisms in the lab. So you'd take the soil that I just showed you, the 1 gram of soil, you'd make a cell suspension in liquid, and then you'd inoculate that into growth medium. And you'd grow the organism in the lab, you'd isolate them, and you'd then classify them. And that's fine for some organisms. It's actually not fine for these organisms. You can't grow these organisms on solid medium, on plates. So they're very difficult to isolate. It takes months to isolate an ammonia oxidizing bacterium. So it's actually, we couldn't do diversity studies of these organisms 25 years ago. But even for other organisms that we could grow easily on plates, we were really worried. And the reason we were really worried was because we were worried that we were just looking at domesticated bacteria, bacteria that could grow in the lab, and not necessarily the ones that were important in the environment. And then 25 years ago, we got molecular methods. So what we do now to study and characterize these communities is we take our gram of soil and we lyse the cells. We break open the cells within the soil to release the DNA, and then we analyze that DNA to get an idea of what's there. So we use various methods, but one key method is to sequence genes within the DNA. And then we compare those genes, and organisms which are closely related have closely related genes, sequences. Those which are distantly related have more different gene sequences. So we can group organisms into closely related clusters or phylogenetic groups based on similarities or differences in their gene sequences. And that's now the basis for microbiome studies. If you've heard the word microbiome, it's become a very popular term, and that's what microbiomics is. It's using these molecular techniques to study microbial communities. And the important thing is that we don't need to cultivate them. Now, what impact did that have? Well, I want to tell you the impact it had on the organisms that we're interested in. I've shown you these four pictures of different ammonia oxidizing bacteria. That's what we thought was the sort of the diversity of these organisms. Maybe 8 or 9, they're not all there, but maybe 8 or 9 or 10 species, and that's it. Well, in the early 1990s, with this guy, Martin Embley, who was at the Natural History Museum then, but we had a very fruitful collaboration together to develop this approach, this molecular approach, to study these ammonia oxidizing bacteria. And what I want to show you on this slide now is a tree of life, if you like, of ammonia oxidizing bacteria that we got using this technique. And what I'm going to show you are techniques that we got, sequences that we got, which are in pink, which were from the very few pure cultures, laboratory domesticated strains that we had then, and then sequences that we got using this molecular approach from a local soil, from seawater off the west coast of Scotland, and from a sewage treatment works in Old Meldrum. Okay. First of all, one group of these organisms, there's the cultivated, the domesticated sequences, if you like, and then there's some sequences from soils, from water, and some more from soil, and then the same for another group here, pink sequences from culture, and then sequences from marine, soil, and sewage treatments, and the sewage treatment works. And the first thing that hits you here, and hit us, is how diverse the community is. Every sequence was different. So every sequence that we got came from a different species. So rather than just having a few species there was enormous diversity. Secondly, none of the sequences that we got from our DNA was the same as any of the domesticated strain sequences. So what was out there was different to what we were growing in the lab. And also we could group these organisms according to environment to an extent. So there was some selection for different groups of these organisms in different environments. Now, to try and convey how amazed we were at this, if you can imagine you're a bird ecologist and that everything that you think you know about the ecology of birds is based on domesticated birds. So you know about canaries and budgies and parrots and so on, and you think that birds out there in the environment are the same. And then imagine someone comes along with a new technique, and that new technique is to go outside and open your eyes, maybe even use binoculars. Imagine how that would change your view of bird ecology. Here you'd see vultures, and you'd see sparrows, and you'd see seagulls and penguins, don't even fly. And you quite quickly get an idea about their ecology because you can watch what they do. You can see some eating worms, some eating berries, some living in nests, some living in on cliffs, and so on. Well, we saw all this diversity, but we can't follow these cells round. So it's much more difficult for us to find out what they do and find out about their ecology. So we and lots of other people then started developing and using techniques to try and find out the function of these different organisms, as well as without cultivating them. Okay, so using these, again, molecular cultivation-independent techniques to try and find out what they were doing. And to ask the question, so what? Okay, there's all this diversity there. Well, does it matter? Do we need it? Do these differences in communities does it make any difference? And one of the basic questions we had was to do with what we called the paradox of nitrification in acid soils. And let me explain what that is. So if you map the world's soils, you'll see all the red soils there are acidic. All the blue soils are alkaline. So you have all these acidic soils, and a lot of those are arable soils, so a lot of those soils are being treated with fertilizer, and that fertilizer is being washed away if these organisms are converted or being converted into greenhouse gases, if the plants don't get the fertilizer first. Now that's not necessarily a problem, in that if you measure the rate of nitrification in soils with different pH, different acidities, you actually find there's not that much relationship. So it's not like pH affects the rate of nitrification. But these organisms, if they grow in culture, they have a problem, and this is the problem that they have. Here we have an ammonia-oxidizing bacterium cell growing in liquid medium with ammonium. And what happens is the ammonium equilibrates with ammonia and with protons, with hydrogen ions, which determine acidity. The ammonia goes into the cell. When it's in the cell, it's converted into nitrite, and the nitrite then leaves the cell. And when the nitrite leaves the cell, it equilibrates in the liquid with hydrogen ions to form nitrous acid. So that's fine under normal conditions, neutral conditions, non-acidic conditions. But if you increase the acidity, you increase the hydrogen ions, then you get more hydrogen ions. That pushes the equilibrium over to that side. You get less ammonia going into the cell, less nitrite produced. And what nitrite is produced is mainly there as nitrous acid. So under acid conditions, These organisms have two problems: hardly any ammonium substrate and lots of nitrous acid inhibitor. So that if you isolate these organisms and grow them in the lab and look at the effect of pH on their growth, you see this, that they only grow at neutral or alkaline pHs. They don't grow as soon as you reduce the pH below 6.5 or 7. And this includes the red strains there, those nitrous amonis strains and nitrous aspidus strains. They're organisms that we've actually isolated from soils with very low pH, pH 4, pH 5, but they can't grow in this medium at these low pHs. So this is the paradox. The organisms, these ammonia oxidizing bacteria, can't grow under acid conditions, but you get very high rates of nitrification in acid soils. Now, to answer this question, we needed three things to do with Aberdeen. First of all, Zena Smith was a technician who ran my lab for many years and grew these organisms. And without her, none of this work would have been done, because it's not easy to grow these organisms and keep them alive and keep them pure. The second thing we needed, well, that was very useful at least, was this field. And this field is near the airport. It's part of the Cravestone pH control plots, part of the Scottish Agricultural College, as was plots. And somebody, and I am eternally grateful to them, in the 1960s decided they wanted to look at the effect of pH on crop growth. So they set different plots in this field at different pH values. And those pH values have been maintained annually, twice annually ever since. So we have plots of soil which have been kept at pH 4.5 and 5 and so on up to 7.5 for more than 50 years now. So this provided a great experiment, a great experimental site for us to look at the effect of pH on our ammonia oxidizers. And the third thing we needed was this guy here, Graham Nicol. And Graham Nicol actually came to work with both Anne and me. And Graham was interested in archaea, and there's another great story which I don't have time to tell you about archaea, because there are two, I'll just tell you a brief bit of it, there are two forms of life, bacteria and archaea. And we have in fact evolved from the archaea. We are just a branch of the archaea. But people used to think archaea were scary things that only grew at very extreme temperatures or salt concentrations and so on. But we now know that they grow everywhere. And what was even more exciting for us, and scary in a way, was that we've also discovered, well, somebody else discovered, but we've followed it up, discovered that they oxidize ammonia. So there was then this big question which Graham wanted to answer. Are these organisms, these archaeal ammonia oxidizers, more important than bacterial ammonia oxidizers? And again, to give you an idea of to try and convey the scariness of this, imagine you think you know about everything that can fly, because you know all about birds, and then you discover insects. Well, that's what this was like for us, finding these other organisms belonging to a completely different kingdom that could do this same process. And what Graham did when he left Anne and came and worked with just me was develop techniques to look at the activity, molecular techniques to look at the activity of these two groups of organisms, the ammonia-oxidizing bacteria and the ammonia-oxidizing archaea. And he looked at them in their activity in these soils. And what he found was that as the pH increased, as it went from 4.5 up to 7.5, the archaeal activity decreased. So they were most active at the low pH, in the low pH soils, whereas the bacteria did the opposite. So this suggested that the archaea might provide the answer to this paradox. And again, I'm cutting a long story short with lots of people, but what we then managed to do was to isolate this organism, Nitrosatalia divanaterra, from this acid soil in this Cravestone site. And there it is there. It's about 2 microns, a bit less, 1 to 2 microns long, a fifth of a micron wide. And, okay, so what's so important about this? Well, it's an archaeal ammonia oxidizer. And whereas our bacterial ammonia oxidizers grow at those pH values. This organism grows at that pH range. It only grows at the low pH values, only grows under acid conditions. And then, okay, so this is what happens in Cravestone. What relevance is that to the rest of the world? Well, work was done by Laura Leite-Verta-Morley, another postdoc then in the lab, Cecile Gubri-Ranjan, who was not only in the lab then, she's also in the audience today, and now has her own research group. She did a global study to see where the genes from these organisms were present, which soils they were present in. And she found that one group was present only in acid soils. You'll be pleased to know I don't have time to describe those data, but the red ellipse there is one group of ammonia oxidizers or genes that were only found in acid soils across the globe. And Nitrosatalia divanaterra belongs to that group. Now, just to finish off with an Aberdeen angle to this, when Graham found those first results, people actually said to us, oh, well, it's obviously something strange about that soil, strange Scottish soil. And I found that quite amusing because I'm English, but Graham's not, and he's Scottish, and he was quite annoyed. And that's one reason why we did this study, and why we were really pleased to find out that this strange Scottish soil was giving us the answer to this global paradox. And that name there, Nitrosatalia divanaterra, sounds fancy, sounds sophisticated. Nitroso means ammonia oxidizer, talia means rod, so it's an ammonia oxidizing rod. What about divanaterra? Well, terra means soil, and divana is an old Roman name for Aberdeen. So it's an ammonia oxidizing rod from an Aberdeen soil. So that's my last bit of local interest in this nitrification story, but I'm sure there'll be many in the future. So thank you very much.
Jim, thank you very, very much. That was absolutely brilliant, and just demonstrating not only fantastic Aberdeen brains, but Aberdeen soil as well. Colleagues, would someone like to ask a question? Please, Neva.
Thank you again, Jim, very much. That was very, very interesting. I always wondered what you did down in the laboratory there, your dirty hands.
I don't go to the laboratory, don't worry.
Why has nature created this enormous diversity of ammonia oxidizers? Is there a reason that you've hypothesized for this huge variation? And can we use it to our advantage?
Well, the first thing to say is this isn't specific to ammonia oxidizers. This level of diversity applies to any group of bacteria that you find in the soil or in natural environments. There is an incredible amount of diversity there. What people have tended to focus on, because all of these studies are really testing evolutionary theory, ultimately, because you're looking for creation of diversity and then selection of different groups. People have tended to focus at the ecological end, where you're looking for biogeography, if you like. You're looking for selection of different groups in different types of environments and trying to relate that to the physiology. The question you're asking is, where does the diversity come from? And there's other people who are studying that, but they're more on the evolution side. And what they find is that you can take this one study where somebody has taken a culture of E. coli and then has subcultured it now for 25 years, I think. And what he's ended up with from a single culture is an enormous array of different E. coli strains. And you find this if another group are looking at pseudomonad organisms, and you can add pseudomonas, a pure culture, clonal culture of pseudomonas. You can add that to liquid medium, and very quickly you'll get lots of different types of pseudomonas, different variants of that strain. So we don't know a lot, we don't know enough, certainly, about how the diversity is created, but it's not just ammonia oxidizers, it's any group of bacteria or archaea.
Jim, thank you. Colleagues, please.
I mean, that's a great talk. I mean, presumably the biodiversity is very important for robustness, but I mean, how is that affected by modern farming methods, for example?
Okay, so well, if we look at just the ammonia oxidizers, Probably not too much. Generally speaking, cultivation reduces the richness, reduces the number of species present. But when you've got several hundred or 1,000 species in a gram of soil, then you can afford to lose some. Plus, these organisms tend to survive very well. Partly, and there's a lot of variation within the microenvironments within the soil. So you may be cultivating the soil and making most of the environments, maybe maybe increasing the pH of most of the microsites, but some of them will still be at a low pH, and the organisms may well be able to survive there. So that's not to say that modern cultivation methods aren't having detrimental effects on the soil communities, but it may not be in the diversity per se. It's more likely to be in other ways.
Jim, thank you very much. And colleagues, I wonder if you would join me in just thanking Jim once again for a fabulous talk. And now last but absolutely very much not least, it's my pleasure to introduce Anne Glover. Anne.
Principal, thank you very much, and thank you for the opportunity to be able to speak to you all today, and also to share the platform with Neil and Jim, which is a great honour. I'm going to talk about something completely different. So although I too am a microbiologist, and happy to call myself, even though I did a degree in biochemistry, and that's just jokes for insiders. But I saw the light and became a microbiologist. And I became a microbiologist when I came to the University of Aberdeen. And I came here in the early 80s, really a little bit by accident, but you don't need to know the background of that. But Aberdeen has captured me entirely for my whole career. And that's not because there weren't other things to do or other places to go. It's because every opportunity I could possibly have wanted was provided at the University of Aberdeen. Can I just check? Can everybody still hear me at the back? Because it's weird for me. Thanks. Okay, so Aberdeen provided amazing opportunity. It provided all the equipment I might ever want to do, all the intellectual stimulation I ever might want to have. It didn't put up any barriers for me and who I wanted to work with, even if it was people from different parts of the university. And it supported me every bit of the way. And you might think that that's normal in universities. And because I have been away a bit from the university and visited many, not just in the UK, but all over the world. And we are unusual, but unusual in a very good way. So when I came to Aberdeen, I came first of all as a postdoc and learned lots of interesting new stuff, actually about slime mold, but I'm not going to tell you anything about the slime mold. We've had weird and wonderful things already. But within a year, I found myself with the title of lecturer and starting my own research career. And that was actually quite a scary thing to do, but in a place like Aberdeen, for the reasons that I've said, not as scary as it might otherwise have been. So one of the things I was interested in was not Jim's work, although of course I am interested in that, was, and not also traveling, although of course I am interested in that as well. But I was very interested in how inside a cell, and all of us are made up of billions of cells, but inside that cell, there's huge numbers of proteins, and they all do different things. They've all got different jobs of work. But they only work if they're in the right place at the right time. And I was quite interested in how things got to the right place at the right time, because it seemed like, to me, a very big problem. And in pursuing that, I went with my first PhD student, Brenda Winning, to a conference in the Algarve. And so this is where we went. It was Albufeira, and so this marvelous place here. And for those of you who've not been to a scientific conference, I'm going to really shock you, but we do not work all of the time we are there. So sometimes when you have a conference dinner, after the dinner you might go with the new friends that you've met, and if you're Scottish and foolish, then you go swimming at night, even though it's April and nobody else, but particularly nobody Portuguese, would ever go into the water in April. But we thought it was very warm. And so my PhD student and I both went in swimming with a whole bunch of other people. And we were swimming around, and she stood up from the water. And I'd never seen anything so beautiful in my life. Her entire body was shimmering from head to toe. So she was illuminated. And I just, wow. And I stood up, and she went, wow, I have never seen anything. You get the story. So we were both shimmering away. And so this was nothing to do with the topic of the conference, but I couldn't get this out of my mind. The fact that there was these organisms, biological organisms, which emitted light. Now, we've all heard of fireflies. They're an obvious thing which emits light. But I'd never come across this marine bioluminescence. And so that conference for me was a turning point, because I couldn't get it out of my mind and wanted to find out more about it. So, in fact, we did know a lot about how organisms emit light. And many people had cloned the genes, the genetic information, that make the proteins that emit light. And so one of the questions that I started wondering was that I wonder if you could take the genes from one bacteria that emitted light and put it into another bacterium that we might be interested in for other reasons, such as the bacteria that Jim has just been talking about. So those bacteria that might grow in soil. Because as Jim has just told you, it's terribly hard to culture a lot of these organisms, and we can't follow them, we can't track them, we don't know where they are and what they're doing. So I wondered if we could put bioluminescence into such an organism, would it then glow in the dark? And could we follow it in the soil? So we took the genes, we put it into a bacterium, and then turned the lights out, and hey presto, we now had a bacterium that glowed in the dark, and we could follow in the environment. And this for us was quite a fantastic, kind of wonderful moment. And if we want to have a look at them, then here's a culture just taken in normal illumination like this, turn the lights out, and here are the bacteria glowing in the dark. So we started asking some questions about what do you need for the bacterium to glow? And actually this work was done in collaboration with Jim. And one of the things that we were interested in was seeing if we could take a bacterium that glowed and follow it in the environment. But we found some unusual things, some of them being that the bacteria didn't always glow the way we wanted them. So sometimes there was very little light, and then all of a sudden the light would increase, and we couldn't quite understand what was happening. And this turned out to be a very interesting phenomenon, and I'm mentioning it because talking about it does me no credit, but it's just to tell you how amazing science is in a way. In looking at this work, as I say, I found it an irritation that a single bacterium didn't give out a constant amount of light. So in order to achieve that for our purposes, I thought what I would do is take the genes and take away the bit that controlled how much of the genes were made into the bioluminescent protein. Because I thought by doing that and having the genes switched on, fully on all the time, it would solve the problem. And it did solve the problem. But what it missed, and the question that I wasn't asking, was why is there a system that only when the cells are at quite high density do the light switch on? And actually this turned out to be a major phenomenon, unfortunately not described by me, but described by one of my colleagues, Gordon Stewart, who was at the University of Nottingham at the time, and by coincidence had been a postdoc in the lab when I was doing my PhD at Cambridge with him. And that turned out to be a mechanism whereby bacteria, these tiny little things, no nervous system, no ability, no consciousness like we have, but they have a way of telling how many other bacteria just like them are in the room. And that's really important for a bacterium, because if you're on your own, you won't pick a fight. Okay, so they're a bit like humans in that respect. But if you've got lots and lots of friends with you, just like you, then you might pick a fight. And that's what bacteria do, particularly when they are mounting infections. So they will never mount an infection if there's just one of them, but how do they know there's only one of them? Because they use this signaling mechanism, which is tied up in how bioluminescence is controlled sometimes in order to do that. So that a slight diversion, but what the bioluminescence tells you is, for us, we can use it to find out what genes are active by putting the bioluminescence genes downstream of a gene that we're interested in, and we can find out when the bioluminescence comes on. It tells us when the gene is switched on. It tells us when the bacterium that the genes are in, the bioluminescence genes, it tells us when that bacterium is metabolically active. And that's also very interesting, because sometimes you really want bacteria to be active to do jobs for you. And I'm going to come back to that in a minute. And it also tells us what affects microbial activity. So there are lots of things which will depress microbial activity and lots of things that will stimulate it. And again, that's often very useful when you're trying to manage processes that bacteria are involved in. Here's one thing that we found out, which was quite interesting, is we take a little sample of bioluminescent bacteria and we add an increasing amount of a toxin. So something that might be toxic to you or me, or it could be like a heavy metal or something like that, cadmium or mercury, or it could be something like polyaromatic hydrocarbon or an organic compound. And if you add increasing amounts, then you see the light go out. And the light goes out in proportion to the amount of toxicity. And that turned out to be rather an interesting observation, because along with a number of other colleagues at the university, some of whom are still here, Graham Payton, for example, we decided to set up a spin-out company in order to see if we could use this bioluminescence technology in order to diagnose whether land was contaminated or not. And if it was contaminated, whether we could identify how we might clean it up. Now, that sounds like quite a kind of easy and simple thing to do, but it's incredibly necessary, because with an ever-increasing population, and Jim mentioned 7 billion earlier on, we don't have enough holes in the ground to be able to dig and dump contamination and put it somewhere else. So we really need to think of more imaginative ways to try and bring contaminated land back into use, whether it's for housing or factories or recreational use. And so what we did using these bioluminescent bacteria is developing the technique even more, and I can see Hedda Weitz in the audience, and Hedda was involved in much of the development of this technology. What we wanted to do is we wanted to see whether there were possibilities of detecting contamination in soil by taking a sample of the soil, taking it back into the lab, mixing it with our bioluminescent bacteria, and asking, did the lights go out and how much by? And the short answer is yes, that's exactly what happened. But it would also give us clues as to the nature of the toxicity, because if you're a heavy metal, like I mentioned mercury, then the lights go out in quite a different way than if you're something like an organic compound like benzene or something like that. So you get a very sharp decrease with a heavy metal and a very shallow decrease with other compounds. So it was to some degree diagnostic. And it also told us an interesting thing about bioavailability. So let me just mention that for a moment. Many of you, especially if you're my age, might have one or two fillings in your teeth. And the fillings in your teeth are used to be silver colored, and that's because they were made of a mercury amalgam. And you wouldn't think you would want to put mercury in your mouth, but in the form it's in your mouth, it's not biologically available, so it will not cause you any problems. If you change the form of that mercury, it could be very toxic indeed, so if you vaporize it. So if any of you live downwind, and we've got a southwesterly prevailing wind here in Aberdeen of a crematorium, for example, then a lot of mercury does get vaporized and will flow off in a plume downwind and deposit itself in your gardens when it can be bioavailable. And I'm being a bit mean here because the concentrations we're talking about are absolutely tiny, and so it's nothing too much to worry about, except to say it does matter whether something is biologically available, where it causes problems, or not, where it won't cause you any problems at all. So using this technology, we could do a map of land, and here is a 3D map of a piece of land looking at, in this case, red where the land is very toxic, green where there's not much problem, and the yellow outline indicated where we might, at which point toxicity went from being dangerous to a problem that we could effectively clean up. And so you had a technique here which was actually fairly revolutionary in its ability to be able to clean up, identify, diagnose contaminated land, and also develop options for cleanup. As well as bacteria bioluminescing, so do eukaryotes. And so here's the firefly that I mentioned earlier with its lantern tail. Male fireflies flash their tails to attract females. That's what's going on here. And so we could also take the genetic information which generates bioluminescence in more sophisticated organisms like flies compared to bacteria, because the genes are different. And what we wondered is whether we could use this luciferase to measure different things, like the amount of energy in a cell, to identify compounds that are toxic to us that might not be toxic to a bacterium, and also to see if we could use it as an alternative to animal testing. And so here we put the bioluminescence now into a small nematode worm. So this is just 1 to 2 millimeters in length, but huge compared to a bacterium or many fungi. So here we have the genes. We put it into our worm. This is a microscopic imaging of a worm. And we found that just in the same way as bacteria give out light in proportion to the amount of energy they had inside the bacterial cells, so did the worms. So they gave us the same amount of light. And this work was carried out largely by Christina Legido and Debbie McLagan in my laboratory. So this has a number of applications in drug discovery, partly because the way energy is generated in our cells is largely associated with the functioning of a small compartment within all of our cells called the mitochondrion. And it now appears that mitochondria are very closely linked to a number of pathologies that many of us will suffer from. And indeed, aging is partly to do with mitochondrial misfunction. So what we wondered was, using our bioluminescent worm, whether we could test drugs or potential therapeutic agents to improve the function of mitochondria on our worm, to be able to provide an alternative to animal testing and to speed up the development of new drug discovery. We were able to do this, and we were also able to identify a number of interesting genes which have implications for a number of diseases where mitochondria are closely implicated. So, for example, we looked at genes such as myopathies, where your muscles aren't working properly, many diseases of metabolism like diabetes, or actually also neurodegenerative diseases such as Parkinson's disease and Alzheimer's. And so this technology allows you a different way to be able to interrogate what genes are important in predisposing you to these diseases, and also what possibilities there are to identify new drugs or treatments to be able to treat some of these very difficult or intractable diseases. So that's part of the research story, but scientists are just almost impossibly curious people. That's all we are. We're just, I always think everybody is born a scientist. It's just that most people have it knocked out of them, and the rest of us, it just stays with us all our life. So we're always asking why, when, where, what. And I've certainly been like that. I'm just curious about everything. Everything amazes me. No more so than at the moment where you'd have to be crazy not to be amazed generally by what's going on in our world. But part of it for me was thinking, how would the knowledge that we generate as scientists be useful for everyone? Well, I showed you, and in fact both Neil and Jim gave really good examples of taking the knowledge that we generate in the lab and translating it to have real impact for people and environments. And one of the things that I wanted to do was to see how we could use the knowledge generated from research to help better policies being made that affect all of us, whether that's policies about how we look after our environment, how we have cleaner energy, whether we have safer drugs, whether we have more sustainable agriculture. Any of these questions, they all require knowledge and input to be able to come up with sensible policy. And so that meant me leaving the university or a secondment to Scottish Government as the first Chief Scientific Advisor for Scotland in 2006. And I was there for 5 or 6 years and had the most fantastic time trying to persuade people not that comfortable with science, I would have to say. So I think most people thought scientists, or most people in government who I met initially, thought we were to be kind of feared and avoided at all costs. So you could see people spotting you in the corridor and then dashing into a room. But I'm quite persistent, so I just followed them and told them what I thought they needed to know. And actually it was a wonderful experience, because when you explain to people the power of knowledge and how it can really work, and I can remember saying to one Scottish politician, who I will not name, of course, but this person was very recalcitrant and wasn't really keen to listen a lot about science, engineering, and technology and its value across the whole of society. And eventually, I thought the only thing I can think will work is I said, you know what? Science will actually make you look good. And from then on, we were the best of friends. But when I left my role as Chief Scientific Advisor for Scotland, I was lucky enough to be offered the role of Chief Scientific Advisor to the European Union, or to the President of the European Union, who was then José Manuel Barroso. And this is another amazing thing about Aberdeen University, because although my CSA role in Scotland was part-time, when I discussed with José Manuel Barroso about working with him, I said, well, maybe I could work part-time, and he said no. And he said for him, that's a deal breaker. It has to be full-time or nothing. So I thought, well, okay. And so I came back to the university and I spoke to the principal. And I said to him, so I'm going to resign from the university because I'm going to take up this job in Brussels. And he said to me, why resign? And I said, well, because it's a full-time job. And he said, no, take leave of absence. Don't resign. And I was a bit shocked by that because I'd made up my mind I was going to resign. And I thought, you know, I could see what was in it for me because, in fact, what that gave me was a wonderful power of always being able to do what I thought was right without any worries at all. And all of us have them, personal worries about, oh, you know, how am I going to get another job very quickly or whatever it might be. I never thought about that because I always knew I had my university behind me. And it was a wonderful thing. And I can remember, Ian, you saying to me that if universities don't support people to do things like this, then who will? And that's a great thing to think that universities will do that. And I'm proud to be part of a university that's demonstrated that it does do that and continues to do that. So in going to Europe, Ian also said that we in the Northeast are not very good at boasting about what we're good at. And actually in Europe, we're not very good at it, I would have to say. Europe is #1 in the world in terms of science, engineering, and technology, the impact of that. If you went out into the street and asked someone, I bet my bottom euro or pound, they would say, it must be North America, surely, it must be the US. But it isn't, it's us. We do fantastic science, and so we should get best use of it. And my role in Brussels was to try and communicate how we can use the knowledge that we generate from our science to the positive impact for all of our citizens, and not just for European citizens, for global citizens. This often came into my mind. This is one of my favorite films. It's called Lost in Translation, Scarlett Johansson and Bill Murray. And they weren't quite communicating with each other. And why it was often in my mind is because I was often with Jose Manuel Barroso thinking, we're not quite communicating with each other. But my job was to try and persuade him how valuable science, engineering, and technology was, not just economically and socially, but also as instruments of government to be able to be sure that we got the smartest possible policy for what we wanted to do. And so there's a whole story behind the wonderful time I also had in Brussels, where people really did listen to the fact that evidence must be used in policy, but were also quite clear about the fact that sometimes evidence is not the only thing that matters. And scientists sometimes get too hung up on this and think if the evidence says this, we must do that. And there are very good examples of where the evidence says you must do something, but none of us would ever do that because we're human and we have different values and different interests in life. So just to confirm the last thing about communication, I would argue to all of you that scientists do and certainly should have a really important role in ensuring that many of the policies that affect all of us are evidence-based, because that should at least be the platform on which we start discussing things. And we shouldn't cherry pick. All of us have our own philosophies, but you need to be open-minded. You need to look at the evidence, and if you don't disagree with it, don't blame the evidence. Then you need to explain why you don't want to use that evidence. That's the best way to go, and that, for me, would deliver really impressive politics. I put this up because Douglas Adams, another good author who wrote The Hitchhiker's Guide to the Galaxy, he had this wonderful invention, the Babel fish, which you could take and you could pop it in your ear, and it was a universal translator. So when you had it in your ear, you could understand anyone from anywhere in the universe who was speaking to you. But he also said the downside of that is he thought that the Babel fish was possibly the one thing that had been the cause of more wars than anything else in the universe, because people could actually understand what each other was saying, because sometimes it's better not to know. But anyway, I leave with this idea of communication, because I remember my PhD supervisor saying to me when I was being difficult and not doing something he wanted me to do in terms of communication, He said, remember this, that research not communicated is research not done. So always communicate what you do, whether it's to another scientist, whether it's to the public, whether it's to a politician or a policymaker. You need to communicate what you do. And I haven't forgotten that, and I've tried to do that. So with that, I thank you very much indeed. Thank you.
And thank you very much. That was absolutely brilliant. Now, who'd like to ask a question? You can move from the science to the politics, anywhere in between. Please, sir.
Sorry, Anne, I know this is a really big and probably naive question, but when we get hardly Brexited, how is that going to affect the sort of things you were talking about in the second half of your talk there, particularly in the British context?
Duncan, I don't know. There is a problem. Science, one of the reasons it's so attractive to me, and I think most other scientists who did not vote for Brexit, the vast majority did not, is that science is a universal language. Wherever you can go anywhere in the world as a scientist and work, and we collaborate very well, and indeed we get proportionately much more out of the EU in terms of research funding than we contribute. So we get a very large amount of money. The UK government have said that they will try and compensate for that by making available an additional £2 billion of research funding. But it's unfortunately, that's not the simple answer to this, because you know, I guess if I asked people around the audience to put their hands up who was born in the UK and who was born outside the UK, I think we would find that, you know, in our university, which again, one of its wonderful things is it's so international, we need to attract the best talent, and we become not very attractive. I think the UK is already demonstrating, in people's minds, it does not seem an attractive place to come. So we'll find it hard to get people to come here. A lot of our own people will try and go elsewhere to follow other funding streams. And collaboration is the thing that the European funding system delivers, which is the most wonderful thing, so that I can get a research grant, and Neil was talking about working right across Europe in consortia, but I can work with somebody in Estonia, somebody in Germany, somebody in Portugal, and somebody in Italy on a grant. And I just write the one grant, we all write it together and we put it in. Whereas even just working with one person in the US is quite a challenge to do, because we both have to apply to different places for funding, and if one of us doesn't get the funding, we can't do the joint grant. So I am always optimistic about most things. There's only two things that I'm not very optimistic about. One of them I'm not going to tell you, and the other one is Brexit turning out well.
Has anyone got a slightly happier question? Please, Ferdinand.
Thank you very much, Ian. I'm a bit of an interloper here, so I apologise for that. But a question on science communication, which you've raised and which I think is really important. If I can, without wasting too much time, tell a short anecdote. Before I came into my present job, I was president of Dublin City University. And in that role, we set up something called the National Institute of Cellular Biotechnology in Dublin. And as we were seeking planning permission for the building that was to house the institute, we had significant objections from some of the local population. And it turned out that the reason for the objection was that they thought we were going to clone humans in this particular building. Now, without going into cellular biotechnology, which I don't know enough about anyway, clearly that wasn't part of what we were intending to do. But the particular suspicion took root, and there was a huge campaign, it has to be said, supported by the local Roman Catholic parish priest, and it went into really strange directions. And just as a quirky aside to it as well, we were actually at the process at the time of poaching an individual scientist from Imperial College London, who was called Oliver Dolly, who was a neuroscientist, And so we managed to persuade the local population that we weren't going to clone humans, and the local newspaper ran a little article with the heading, "No Dolly for DCU," which was obviously a reference to Dolly the sheep, but our colleague Oliver Dolly had just come over, and the first thing he saw in my waiting room was this headline on the newspaper. Anyway, I apologize for that lengthy story, but my question is this. In different contexts and in different places, we're not always as good as we should be in the academic community in communicating what it is that we do, why we do it, what the benefit is, what politicians need to do to respond to that. You've played a very critical role in that. Do you think that across society more generally there's more that could and should be done to get that right?
Well, of course, yes, of course I do. Although it is really difficult for us, and sometimes I think we beat ourselves up too much, particularly as scientists, because people tell us we're very poor communicators. But in all walks of life, there are poor communicators, and where anybody has a specialism, they often dive into the specialism and don't just speak in plain language that all of us can understand, and they do it for a number of different reasons. I think one of the things, again, this sounds like an advertisement for our university, but we have a unit, the Public Engagement with Research unit at our university, and they do a great job in working with researchers to try and identify how you might be able to communicate what you do, and mostly not to other scientists, because we have that kind of taped, we know what to do there. But it's doing it more widely to try and identify the impact. So, I mean, one of the things that I always think for a researcher is that not every researcher wants to set up a company or go and talk to big business or whatever, but they should know who would be interested in the knowledge they're generating. And they should have some mechanism to be able to at least pass that on to someone else and say, this would be really valuable for those people, whether it's somebody in agriculture or air quality or whatever. And there should be good ways of doing that. But I would also urge all of those who are not scientists to be smart consumers of our knowledge, because they are often resistant to knowledge because it's sometimes very inconvenient. And actually, if I may, just one thing, I'm taking your lead here and telling a little bit. There was an Ipsos Mori poll in 2016, just last year, and it was looking at trust in society. And right at the top, there was doctors, teachers, judges, then scientists. So right at the top with a very high level of support. And right at the bottom, unfortunately, were politicians and journalists. Now, that just doesn't bear thinking about, because we get most of our information from journalists who we don't trust, and we elect people to represent us in Parliament, and we don't trust them either. And I'm thinking, why don't we trust them? Because most people go into politics, they're well-meaning. They want to do something for all of us, and not everyone does. So that's a good start. But it's often because they feel, for whatever reason, and this might come back to the journalists, I don't know, but they will not speak straightforwardly and honestly about things, saying, I know the evidence says this, but we have decided to do this for these reasons, and let me take that through. Often, I've heard people, they will blame the evidence, say we're not going to do anything at all because the evidence isn't certain yet. But just my very last word was, as a chief scientific advisor, the UK have had chief scientific advisors for a very long time, and Winston Churchill once said that what he most wanted in life was a one-handed chief scientific advisor, because the problem with science advisors is they're always saying to you, well, on the one hand, and that, of course, is a problem for scientists. We love uncertainty. Politicians, rightly, dislike uncertainty. So how do you find the middle ground? And that's the real challenge. It's a challenge worth stepping up to, and we could all do better, I think.
And thank you very much. And colleagues, thank you very much. I know everyone came this evening expecting our 3 talks to be wonderful. I don't think any of us realised how wonderful they would be. So, Neil Gow, FRS, Jim Prosser, FRS, and Glover, FRS, please join me in thanking them for the fantastic presentations. And that concludes this evening. Thank you very much.